Hello, General Botanist. It's me, Stephanie, and we're going to be talking about seedless vascular plants. You'll be seeing this acronym SVP a lot. Let's get into it. So here are your learning goals. We're going to know the position of the seedless vascular plants within the larger plant phylogeny. We're going to know the short list of characters which, in combination, identify the seedless vascular plants, which are these three groups, the ferns, lycophytes, and horsetails. We're going to be able to explain how all of the new vegetative characters function together to allow vertical growth, and explain why vertical growth evolved. And by vegetative, I really just mean non-reproductive. And then we're also going to know the difference between Homospory and Heterospory, and the examples for each, namely Phylogenella, which is a genus, and the ferns. Sorry, I said that backwards. Here's your vocab list. Notice chapter 17 in the really 6th, 7th, or 8th edition. We'll get you most of the definitions here and some of the figures that you'll be seeing too. Okay, so we're talking about plants that evolved from 420 to 360 million years ago. And this is especially when they were really the dominant organisms on Earth. Once plants were able to sort of grow vertically, they kind of took over, and they were able to grow really, really big, right? And since they were so big and they were so dominant on land, and they really didn't have any other, they didn't have much competition. Herbivores weren't around yet, for example. They really did a number to the environment. They basically oxygenated the environment and took an incredible amount of carbon dioxide out of it and turned it into carbon. And this carbon that these early forests of these seed-free vascular plants, the carbon deposited from these forests is actually what we burn for coal. So it has come full circle, and really that's a lot of what we used to burn coal. It really is these, especially this one genus, Lepidodendron. This was a very tall tree. It's sort of you can see the fossil here in the picture on the left. This is an imprint of basically the outside of a very tall tree. Now, they didn't really produce wood. Now, the seed-free vascular plants, they're vascular, but they don't produce secondary xylem. But they were able to still produce trees that were, extremely, extremely tall, like you can see in this picture. Cool. Wow. Similar to the sort of habit of a tree that we're used to, but very different structures, as we're going to be talking about. Now, all of the living organisms of these groups, sadly, sorry to burst your bubble, they're not this big. All of the ones that were this big are indeed extinct. We have lots of cool ones, though. They're just much smaller. And the ferns are a well-known example. So here's that. Okay, so we talked about the bryophytes, and the last one I just sort of have these three arrows representing the hornworts and the mosses and the liverworts. And now I've expanded on the seed-free vascular plants, the SVP. And we can add a couple of things actually that we've sort of talked about. Well, they're vascular, and so by that I mean they've got phloem and xylem. Specifically, they've got lignified xylem. Lignified tissue, meaning tissue that's sort of hard, and it's going to allow them to sort of keep their structure even when the cells die. Remember those sclerenchyma cells? And so so once they're able to form this vascular tissue, they're able to sort of differentiate their body parts into the different organs that we talked about earlier in the semester, the roots and the stems and the leaves. So this is when they evolved, right? The leafy things we talked about in the mosses, those weren't actually true leaves, and this sort of has to do with their venation, really. The fact that they didn't have real venation in the bryophytes. Okay, so let's add some of those terms. Xylem and phloem. In other words, vascular tissue. We've got the presence of the compound lignin to provide rigidity to cells. We've also got some things, sort of remnants of the previous video, the fact that we've got actually a switch in the dominance of the major parts of the life cycle here. We now have sporophytes as the dominant part of the life cycle. Interesting. The sporophyte will also be branched. This is another feature of seed-free vascular plants. And actually, all of those 3 hash marks that I just added to this cladogram, not only do they apply to the taxa we'll be talking about today, those ones I just highlighted in red bracket, but they also apply to the rest of the descendants, right? Namely, everybody else from this lineage, namely the seed plants. Yeah, it also applies to them. All of those things. But of course, the seed plants have other features, and we'll get into those another time next week. Okay, just a couple of cool pictures of some plants we'll be talking about. Here's a fern on the left, and a horsetail on the right. The horsetail on the right is actually showing 2 different life life stage techniques here. Now they're both actually diploid structures. This is all sporophyte, but this one is producing spores and it's non-photosynthetic, while this one is photosynthetic and it's not producing spores. Cool. Horsetails do that sometimes, not always. Just a fun fact about the ones that actually this picture was taken in Arcata, and so you might see that in the early spring. Just some more kind of cool eye candy of some well-known seed-free vascular plants, and ones we'll be talking about. We're really not going to be talking about this seed-free vascular plants in evolutionary order today, honestly. We're going to be talking about them sort of in order of convenience, and by that I mean like in order of what's most well-known. What you see here we've got 5 pictures. First of all, sad to say that Psilotum, this genus, this is the whisk fern group of seed-free vascular plants, and we're not going to talk about them, actually. Sorry, you're going to have to learn about them on your own if you're interested. And really, these 4 names, these are all genus names, okay? So these genera, they're all in sort of their own respective family, and actually, most of these are in their own respective phylum, except for, well, these 2 are in one phylum. More on that in a minute, but just so you know, these Equisetum over here, this is the genus of horsetails. Polypodium, this is the genus of, well, polypody ferns. There are many, many different genera of ferns. I'm just using Polypodium as one example. Lycopodium, this is our genus of club mosses. Selaginella, this is our genus of spike mosses. We're going to start off by talking about the ferns. I'm just doing that because they're the most well-known, not because they're the most ancestral. The big leaves that you're seeing here, this is all sporophyte tissue. So what we're seeing is a dominant sporophyte instead of a dominant gametophyte, starting with the ferns and starting with the seed-free vascular plants. And the leaves, do they remind you of sort of a term that we talked about earlier in the semester? Where you've got, and this is one big leaf, by the way. It's got this one elongate axis, maybe called a rachis, and then it's got a bunch of these leaflets all coming off of that axis. Yeah, this is a pinnately compound leaf. Yeah. In other ones, like this one back here, it looks like we have a 2 or 3 times pinnately compound leaf. In other pictures, though, like this one, maybe it's not. It is also. It also does look pinnate. Sometimes they're not. Sorry. More on that in a sec. We have a very wide variety of morphologies of sporophytes in the seed-free vascular plants. But the consistent feature is that it's always the sporophyte that's dominant. That's sort of the main photosynthetic part of the life cycle. And it's going to be having usually it's going to have many, many, many sites of meiosis. And now conveniently, those sites of meiosis are still on a sporangium. So we're going to see that term used again. Okay, the pteridophytes, like I said earlier, we're seeing true roots and stems and leaves for the first time. True roots and stems and leaves. And this is because of vascular system has evolved. We've got those lignified cells, namely here we've got those tracheids. Remember, those are part of the xylem, and actually remember they stain red, like they're represented in red here because they stain red with fluoroglucinol. This is a very well-known sort of dye that's used for, well, it's not that well-known. It's well-known in Botany 105 lab because we use it to dye stem and leaf and root tissue to see sort of where the xylem is. It stains red. Cool. Anyway, since they have a way to get water up, to transport water, well, they're able to grow up. This is sort of their way to potentially get closer to the sun, which is something that they would have needed to do early on in their evolution, right? Sort of in between, in this evolutionary stage, in between the bryophytes and the seed-free vascular plants. Well, at some point, everything was sort of the same height. And so everything that's below that is going to get shaded out. And if something was able to be taller than most things, well, it's going to have a huge competitive advantage, and it's going to be selected for. And so, yeah, shading might have been one of those selective pressures that led to vertical growth. Like I said, we have a huge variety of morphologies within the pteridophytes, the ferns specifically. And notice that ending ophida, just like Bryophyta and Hepatophyta and Anthocerophita, that's a phylum name. So, I'm telling you that Pteridophyta is a monophyletic group. Here's the sad truth, and this is sort of hard for us to all grasp right now. Actually, not all ferns might not be a monophyletic group. Now, we use this common name fern. We kind of throw it around, just like you'll see today. We throw around that word moss. We throw it around because it's a common name. It actually does not imply monophyly. And I want you to know Pteridophyta as a phylum that's a monophyletic group of ferns, but I want you to also just know on the side that there's more to the story that we don't really have time to get into in Botany 105, but I urge you to look into it if you are interested in the deeper evolutionary relationships between the taxa we're talking about today. We're really making good generalizations about these taxa for our purposes. Anyway, the ferns, more on the Pteridophyta, one group of the seed-free vascular plants. Like I said earlier, they are sporophyte dominant, just like all the seed-free vascular plants. And then they have these things called fiddleheads. The fiddleheads are the young fern leaves, the young sporophyte leaves. They're always going to grow in this really characteristic way, where they sort of unravel like a cool spiral. And actually, they have a cool term, the development of, or the way that they develop and unravel from sort of a young to old stage. This is called cercinate vernation, sort of the way that they open in this very fancy spiral fashion. Well, all ferns do it. These are other just cool pictures of ferns. More cool pictures of ferns showing the massive variety of morphologies that we see in leaf shapes and sizes. Now, they do all have stems as well, although the stem might not be very noticeable sometimes. A lot of times the stem is underground, and we would call it a rhizome, if that's the case. And sometimes all of those leaves are sort of coming from this one cluster as opposed to along a stem. So we see a variety of habits as well. Finally, some of their sexual parts now. We've got these clusters of things. Here we've got these sort of circles. And if we look really close, there's actually a bunch of little tiny things within that circle. Okay, well, in this other picture, we've got these like really thick margins, right? These thick margins right there. Those are leaflet margins, maybe. Oh, over here, we've also got these little marginal circles. And we've got all sorts of structures. Well, these are called sori. Notice I say these, plural. One of these is a sorus. So let me just label this one that I've circled right there. Oops, just that one. That is a sorus. Together, these two are sori. Okay, and or like this one linear one. Maybe that's a sorus. I don't know. Sort of hard to tell what the distinction is between sori and that one. Well, what are sori? Sori are clusters of sporangia. Yeah, remember? Sporangia from the bryophytes. A sporangium is a diploid structure that is the site of meiosis. And here, let me use a different color. All of these little tiny structures all in there. And the ones like that we actually can't even really see, but they're kind of right in there. And they're like, oh, maybe they're like some right in there. Okay, they're so tiny that we cannot see them in this picture. We would have to use a microscope to see them. But I'm going to label them anyway just so that we associate them with their this term sorus, because a sorus is a cluster of sporangia. So a sorus is like, you know, dozens and dozens of sporangia. And that's why we can't see them in this picture. They're too small. So here we go. Now in this picture, we've taken a very thin slice through a sorus. And that's what this whole structure is right here. This is sort of an X-ray view inside of a sorus. So like if we were to take this section like right here with a very fine razor, slice that open. And then dye it and look at it under a microscope. This is what it would look like. So we've got all of these little structures. There's literally dozens of them. And since this is just a little fine section, you know, like a loaf of bread, it's really hard to capture this very, very three-dimensional structure, or rather, all of these tiny three-dimensional structures. And so you can't really see all their bases, and that's why I'm highlighting them. But really, each one of these sporangia that I'm highlighting has this elongate base, and then it has this structure at the top. And then this one you can see really clear on the end here. That structure on the top, that sort of backbone, is what will open up and allow them to release their spores. Okay, so again, we've got this term sporangium, and let me just do it in black. I'll just kind of, it's one of these like lollipop-looking structures, but I just drew a bunch of them. One of these is sporangium. It is within this giant structure that I've labeled here. Like this whole thing is a sorus, to be very clear. And inside the sporangium, we have a bunch of spores, namely myospores, where we have haploid spores developing inside of those sporangia. And here's what happens: that backbone happens to be called a annulus. That's this backbone part right here. It will, when it dries up in the right conditions, it'll sort of open up and release a bunch of these spores. These are myospores. Remember, whenever I say the word myospore, it implies haploid. It is the result of meiosis. Meiosis is happening, boom, right there in that sporangium. Releasing myospores. Cool. Here is a look at their life cycle. Very detailed. Let's start with those spores that I just mentioned. There's those myospores. Notice, we just have this one type of spore. Yeah. It's a spore and it forms one gametophyte. Notice we don't have male gametophyte and female gametophyte. We just have gametophyte. Yeah, neat. But we do still have these terms archegonium and antheridium. Yeah, it's just that this time the archegonium and the antheridium are both present on a single gametophyte. So in other words, this gametophyte right here, it's sort of heart-shaped. It's got all these rhizoids. This heart-shaped gametophyte is bisexual. It produces both archegonia, like right there, and antheridia, like right there, right next to it. Now, hopefully, from its fern's perspective, it won't self-fertilize. It won't it won't have sperm from this very same gametophyte fertilize an egg on this gametophyte, hopefully. And they have ways of avoiding that. We can talk about those later. But anyway, this archegonium and antheridium, these are the same terms, the same things that we already know now from the previous video. The archegonium is the structure that produces the female gamete, the egg. Cool. The egg is retained. Cool. Same story. That egg will undergo mitosis right here. Or excuse me. Whoa, I jumped ahead of myself. That egg will undergo fertilization with a sperm and form a zygote. That zygote will undergo mitosis and form an embryo. Okay, so everything is sort of the same here, except for we don't have two different gametophytes. We only have one gametophyte. Cool. That is actually what I mean by homosporous. So this term up here, let's come back to it. Homo means same. Spore, well, we have the same type of spore. We only have one type of spore. More on that in a minute when I compare it to this other thing called heterosporous. Okay, let me just kind of finish up here because it's really not that surprising. The embryo will continue undergoing mitosis and it will become a sporophyte. The sporophyte will be attached to that gametophyte, right? Because remember the egg and then the zygote, this is all retained in the gametophyte. It's just that in ferns, the gametophyte, it's a thalloid, like this. It's this heart-shaped thing. That's the gametophyte. It's very small and short-lived. It's not going to branch. It's not going to grow. That whole gametophyte is like a centimeter big or so, maybe much less than that even in some species. And it's very short-lived. Yeah. That young embryo is dependent, and maybe even the young sporophyte, you'd say, is dependent on the nutrients of that gametophyte, and maybe it's dependent on the sort of the structure it's providing it, it's attached to the substrate. So here's just another look of a gametophyte. Now we've just sort of removed it from its environment. It's kind of cute. They usually are sort of heart-shaped like this. And oftentimes the archegonia, remember the female part, they're sort of in the center. It's sort of near the cleave of the heart, if you will. And then the antheridia, the male parts, these tend to be sort of scattered all on the outside. And it's right here on the gametophyte that your egg is going to undergo fertilization, and then that sporophyte will grow like a cute little fiddlehead. Yeah, right there. Cool. And here's a real look at a fern gametophyte. It kind of, actually, it definitely looks just like a liverwort, like a little phallus of a liverwort or a hornwort. Cool. Another picture. Nothing new, just better pictures. Same. Now this is a scanning electron microscopy image. This part right here is sort of the cleave of that heart-shaped gametophyte, and then there it is again, sort of that cleave. And so again, oftentimes we've got the antheridia on the outside and the archegonia on the inside. Now you can really nicely see in this picture down here, let me get a different color, that this, oops. There. This archegonium, it's really three-dimensional. And it has the stalk and the venter and the neck, sort of just like the archegonium of what we saw in the mosses and in the archegoniaphores of the liverworts. It's elongate, and a sperm cell from an antheridium has to sort of get splashed up there and swim all the way down the neck to find an egg cell that would be sort of way down there at the bottom. Cool. Okay, moving on. Whoa, moving right along. We are done with the ferns now. New chapter, the new phylum rather. We're going to be talking about phylum Lycopodiophyta. Sort of a mouthful to say, but I recommend saying these words out loud. It'll help you sort of remember them. Lycopodiophyta. This is the phylum again. Whenever I say phylum, that implies a monophyletic group. This is a monophyletic group, including the spike mosses and the club mosses. Okay, here's what they look like. Really, it's not that diverse of a group anymore. But this is the group that I mentioned earlier that is a home to the Lepidodendron, that genus of really big, extinct, tall trees that we now burn for coal. And here's its relative. It's extant relative. We don't have big woody structures anymore. We have these sort of these sort of more or less herbaceous, honestly kind of creeping style of habit types of plants. Some of the features of the Lycopodiophyta are more primitive than the ferns. I'm not going in evolutionary order exactly today. We'll talk about that when we get to the phylogeny. But again, we've got a dominant sporophyte. Again, we have stems and true leaves, even though they're small, and true roots that indeed are underground and they're transporting water to the above ground parts of the plants. So Lycopodiophyta, when I say it with the ophyta, that implies the monophyletic group. But Lycopodiophytes just implies sort of, some taxa within that are sort of closely related to Lycopodiophyta or within Lycopodiophyta. And we're going to talk about two members of this phylum. I might call them the Lycopodiophytes. The first one is Lycopodium, this genus right here. The common name for Lycopodium is club moss, or these are the club mosses. There's many, or there's at least a few, there's many species of Lycopodium. Oops. There we go. But they all have this sort of elongate upright structure that's going to be producing spores. So instead of a sorus, like we saw in the ferns, they have this sort of, it's really more like a cone-like structure. And it's called a strobolus. Strobolus really just sort of means cone-like structure. And we see it here. We'll see it in other ones. I'll show you. And it's actually these little tiny little structures in there that are probably producing spores in this picture. Okay. Selaginella. This is another member of the phylum, but Selaginella is a different genus. And it's the genus of spike mosses. So, same phylum, different genus. Notice it looks a little different too. It doesn't have as pointy of leaves. Here the leaves are sort of broader. They're a bit more scale-like. Yeah, that's at least a trend. But Selaginella also has a strobolus. But there's a difference between the strobolus of Selaginella and the strobolus of Lycopodium. A big difference. And this is where I will clarify now the terms Heterosporus and Homosporus. Remember the ferns? The ferns are pretty much all Homosporus, with very, very few exceptions. The ferns are Homosporus, like Lycopodium, as you can see on the left here. In this picture on the left, Lycopodium is sort of releasing spores from its strobolus. That's what all these little thingies are right here. And this is the strobolus. And then this is a zoomed in version of that. And now also in this picture on the right, we've taken a very fine slice with our fine razor and we've dyed it. And so we can really kind of very clearly see all of the important things. And those important things that we can see are, first we can see these repeating units. This right here. These are sporophylls. And then we've got these structures that are sort of, these more or less circles, these sort of shapes that are smaller, and they look like they've got red dye in them or something. They're clearly different from the sporophyll. They're like a packet of something, it looks like. Well, this is the sporangia. Oops. SP. Wow, cool. And then inside of the sporangium, you can barely even see them here. These little tiny, tiny blue things, that's a spore. Namely, a myospore. The site of meiosis is that sporangium on the strobolus. We have a bunch of meiosis happening and a bunch of myospores being released. Cool. Spike mosses. A little bit different. Oops. Selaginella is heterosporous. They don't just have one type of spore, they have two types of spores. They're both haploid, but one of them is much smaller. So notice we've got these tiny little spores over here. You can barely even see my little pen tapping here. These are microspores. Yeah. And then over here, and actually, sorry, let me back up. These are microspores too. We've got microspores in a bunch of these different sporangia on this strobolus. I just labeled the one from that particular one for no reason. Over here, we've got a different type of spore, these larger ones. I've just circled 3 right there. I'm going to label this one. It's called a megaspore. Well, that makes sense. Again, it's bigger, mega, micro, smaller. Great. And we can associate those other terms with them. So instead of calling this structure a sporangium, like we did in Lycopodium, we can call it a megasporangium because it has megaspores in it. And same thing with over here. We can call this sporangium a microsporangium because it's got microspores. And you guessed it with the sporophyll term too. We've got microsporophylls and megasporophylls associated with the respective sporangium. So I guess I'll do it. We've got like these. This must be a megasporophyll because it is subtending a megasporangium. And then over here, I'm not going to write it. You got it. A microsporophyll because it is subtending a microsporangium. Moving on. Where were we? Okay, so that helps a little bit with this. Now we're looking at Lycopodium. This is the one that is homosporous, okay? So here's the sporangium. They've just got the one sporangium, and then there's the sporophyll right there. Little sporangium that they've zoomed in on. And we've got meiosis taking place. And there it is. There's those spores, those haploid spores. The haploid spores, since there's just one type of spore, it's going to just produce one type of gametophyte. Here it is. It's producing one type of gametophyte. And that gametophyte, you would call it bisexual because it's got both male gamete-producing structures and female gamete-producing structures, namely the antheridia and the archegonia, respectively. Just like in the ferns. And so that's why I had that term homosporous next to the fern life cycle. Okay. Really same story here. So the antheridia, they're producing sperm cells. The sperm cells, again, we've got really kind of interesting, funky shaped sperms in both the seed-free vascular plants and in the bryophytes. Just interesting flagella, very interesting shaped bodies. And for whatever reason, that kind of helps them get to, or for some reason it was advantageous for them to have that shape because it maybe somehow allows them to find an egg or to kind of swim to an egg. The egg is retained right there on that archegonium, which is on the gametophyte. And then we've got fertilization happening kind of in the egg, and that egg becoming a zygote. Okay. The zygote will undergo mitosis, become an embryo. Same story here. That embryo will continue going through mitosis, and then that's your young sporophyte. It looks like a blob at first, and then it becomes the familiar sort of branch-stalked thing that I showed you a picture of earlier. And the strobolus at the top, well, that's where the sporangia are. And the sporophylls and the spores. It's a circle of life. It always is. It never ends. Ever. Because to be clear, we're talking about, all the evolutionary steps we're talking about between bryophytes and seed-free vascular plants. It's hard to grasp how long of a period of time this took place over. We're talking millions, billions of generations. And some of these generations, maybe they're taking weeks or seasons or years, depending on the types of plants we're talking about. Crazy. Moving on. Lost my train of thought there. We were comparing Heterosporus to Homosporous. And that previous life cycle for Lycopodium, that was Homosporous. Selaginella is our example of a heterosporous one. And now this one's important. This is our one and only sort of example, at least for today. This is our one and only example of a heterosporous life cycle because we had Lycopodium and the ferns as our examples for Homosporous. Selaginella, it's heterosporous because, again, the strobolus, there's a little strobolus right there on the mature sporophyte. That one strobolus will produce two types of spores. namely megaspores and microspores. Okay, and so we already talked about the fact that those microspores are born in microsporangia, which are subtended by microsporophylls. There's those terms again, microsporophyll. And then the megaspore is found in a megasporangium within a megasporophyll. You got it. Well, now we have, now those are the results of meiosis, right? The microspore and the megaspore are the results of meiosis. And so they're going to undergo mitosis, both of them. The microspore is going to undergo mitosis until it undergoes mitosis a few more times until it forms these sperm cells. Again, goodness, look at the shape of this very strange sperm cell. I don't know the details beyond this, but it seems very interesting, and maybe there is some evolutionary advantage. If you're interested in, feel free to look into it. Cool. Those sperm cells, they will make their way over to one of those, retained eggs. Eggs that's namely retained on the archegonia, of which there are going to be a bunch of on the mature megagametophyte. So there's sort of, I guess, a new term too, a megagametophyte. It makes sense. It's the gametophyte that's formed from the megaspore. And the megaspore is always going to be the one that's going to be producing the female parts eventually. And it makes sense because the gamete of females is larger than the gamete of males. The sperm cells is always smaller. In fact, that's really what defines a male and a female in all biological senses of the term. A male has a smaller gamete. Boom. And so a megagametophyte sort of implies, well, that's where female things are happening. The thing with the larger gamete. Okay, so anyway, that term megagametophyte implies haploid, a structure that is haploid and multicellular, that's going to be at some point producing an archegonium and an egg. So everything else is the same. We've got a zygote, we've got an embryo, a young sporophyte, and the zygote and the embryo and the young sporophyte, these are all retained on that gametophyte. But the gametophyte, again, just like the ferns, it's very short-lived. You don't notice it. You only notice this part of Phylogenella, the leafy, fun part. You don't notice this little itty-bitty, short-lived gametophyte, the megagametophyte. You don't see it very much, unless you're really looking for it. Unless you're being a real botanist and really looking for it. Okay. So here's another one of those little summary slides. Oops. This time the summary slide is showing the terms, and the next one is of Heterospory. So notice Heterospory sort of has this whole other half on the right. Homospory. This is everything we've been talking about, just in words. So I don't think I'm going to go over it word by word with you right now. Come to my office hours if you'd like to do that with me. Here's the other one. Heterospory. Review this, copy this down, make sure it makes sense to you, make sure that you can apply all these terms to sort of that major life cycle, showing all of the detailed stages. Here's sort of another just review slide for you to have for your notes. Vascular plants, they either have Homospory or Heterospory, and now the only ones that have Heterospory, the only one we've talked about so far is the spike moss, which is Phylogenella, that one genus within the Lycopodiophytes. But, spoiler alert, all the seed plants are also heterosporous. So the ones we're talking about next week and the week after, they're all heterosporous. Okay, there's some more definitions for you. I'll let you read them. Our last group of taxa of the seed-free vascular plants are members of this order Sphenophyta, or Sphenophyta. Again, I hear it pronounced both ways. These are the horsetails and scouring rush and some other cool groups. But we're going to use horsetails as sort of our major example of the members of Sphenophyta. They look like this, and this is a picture of one that's actually local to our area that you'll see growing. Like I said, kind of early spring, they'll shoot up these structures on the right, which are going to be producing spores. This is the Strobolus. We've already seen that word, and yeah, that's where we've got meiosis occurring. Here's another look of a Strobolus now in this large central image. We've taken that one. we've taken a very fine slice with a very thin razor, much thinner than what I just drew, and we've looked inside of a Strobolus. And what we see are these kind of T-shaped structures. And again, since this is a three-dimensional thing, it's sort of hard to see the whole thing at once. But I think I can see a good one, at least like right here. Kind of circling this whole structure right here. And notice that what I just circled, we've got this structure and then we've got this structure. And these two structures are sort of being held together by this sort of T-shaped thing. Yeah, there we go. Okay. Well, what I circled in green there is a sporangium. And this time, the sporangium, instead of just being right there subtended by a sporophyll, the sporangium of Equisetum is subtended by is attached to the structure called a sporangiophore. So that's going to be on a slide here in a sec, I promise. And if we look inside of these sporangia, we've got these little tiny dots. We can barely see them. But if we were to look at one of them and zoom way in, this is what we would see. These spores are these circular structures that normally, or at some parts of sometimes, their elaters are wrapped all the way around them, tightly wrapped around, like you can see here. They've got these coiled structures all the way around them. But when the humidity is right, the environment is just right for the perfect conditions for dispersal, those elaters, as they're called, they do this. They separate from one another, but they do it pretty rapidly, and they're sort of able to bounce apart from one another. They sort of stretch and just separate from one another, and they all actually do this at once sometimes. You can like breathe on them under the microscope and see them all sort of bounce around in a way. It's kind of cool. If you ever get a chance to do that, I highly recommend it. And here's that life cycle. So back to that sporangiophore, here's sort of one of those T-shaped structures that itself is holding multiple sporangia. Okay, so a sporangiophore is a structure that's it's holding multiple sporangia. Yeah, just said that. Great. Like there's a sporangium. There's a sporangium. They're like right here. They're on that sporangiophore. Okay. Inside of the sporangia, we've got spores with elaters. Those spores will go ahead and go through mitosis and produce, again, just one bisexual gametophyte that has both archegonia and antheridia on it. Same story. Egg, sperm, fertilization, zygote, embryo. Embryo is attached to gametophyte. Gametophyte is short-lived. Sporophyte is dominant. And sporophyte produces a Strobolus. Right? The sporophyte, it's diploid. The Strobolus is going to allow those diploid cells, or it's a site for those diploid cells to undergo meiosis. Fabulous. And here are your little sort of study resources. Feel free to pause this, download the PDF, do whatever you've got to do. There it is again. Same Selaginella life cycle, Heterosporus. And then there's your example of a Homosporous life cycle. But remember, Lycopodium is also Homosporous. And so is Equisetum. Let's add a little bit to this phylogeny. All right. Just a little bit. Let's see. Let's add like Heterospory. So Homospory appears to be sort of what most of the things we've talked about so far have. And even though bryophytes, they're not vascular plants, they have something more similar to Homospory than they have to Heterospory, because those spores, even though they produce two different types of gametophytes in the bryophytes, at least in some of them. They produce the male gametophyte and the female gametophyte. Those two spores looked the same. And you weren't able to tell the difference between those gametophytes until they produced their gametangia. And so, really, the bryophytes also have homospory. In other words, homospory is the ancestral condition. Heterospory, like we talked about, it evolved once on the way to spike mosses. So if this cladogram is true and based on DNA, which I'm telling you it is, we're going to assume it is, well, Heterospory, H for Heterospory, evolved there. But then, spoiler alert, Heterospory also evolved on the way to the seed plants. H for Heterospory. Yeah. So we might have according to this cladogram, we do have an example of homoplasy here, right? We have the convergent evolution of the condition of Heterospory. Cool. The other thing that we will see on our way to the seed plants is secondary growth. That to the degree, to me, has always meant secondary growth. Or in other words, secondary xylem. Wood. We've got woody growth in the seed plants. Some have also lost the ability to have woody growth, but not all of them. So, I mean, you could add other stuff to this cladogram. If you've got it in your notes, you can add things that are sort of unique to each taxon, right? You can combine this cladogram with your previous bryophyte cladogram that had some more things labeled, and write things like, calyptra, write things like sporophyte dominant. We might have heard that earlier. Right, thing like Saurus, Strobolus. Was there an event where we lost the Strobolus over time? Yeah, sometimes we have events, evolutionary events, where a structure is lost over time. What about sporangium? When did sporangia evolve on this cladogram? These are just sort of thoughts. These are things that really we've talked about indirectly or directly, actually, in some of these videos, but just something that I want you to think about. I want you to do that. Don't just think about it. Add some of those things. Add more things. Go back through your notes and be like, here's something that's unique to just the horsetails, or something that's sort of found in both the mosses and the hornworts. I don't know. If you find things, add them to your cladogram. It's a cool study tool. That is all I have for you today. Hope you have a good rest of the day.